Welcome to this webinar hosted by BioLamina. It will be an exciting half hour ahead of us, and if you have any questions at the end of the webinar, please use the email or phone number that is on the last slide. BioLamina is meeting the demand for substrates that are suitable to be used in production of cells that will be going to the clinic. And we are meeting that demand by launching a new cell therapy grade product line. I will come back to this later on, but now I would like to properly frame this story by telling another story, and that will be a story about laminins and also a story about passion. Let's start about the laminin story. Laminins are present in basement membranes that are surrounding almost all tissues in the body. They are binding directly to the cells via integrin receptors, and also they are binding to themselves and also to other proteins that are present in the extracellular matrix. They are the orange cross-shaped molecules that you see in this slide. Laminins are large, heterotrimeric glycoproteins consisting of an alpha, beta, and gamma chain. And there are 16 different laminin isoforms in the human body. Their naming is based on their composition of alpha, beta, and gamma chain respectively. So laminin 521 consists of an alpha 5, beta 2, and gamma 1 chain. Laminins are expressed tissue specifically, meaning that the different laminin isoforms are combined specifically in the different tissues, also over time during development. Laminins cannot be purified or extracted from the tissues without being completely destroyed and thus dysfunctional. And these tools have not been available before to the research community until BioLamina started to make the full-length recombinant human laminins. BioLamina is all about making laminins, and also all about helping you to make better cells. We believe that you have to recreate the niche that you have in vivo also in vitro in order to get proper cells in the dish. BioLamina is also about passion, a passion which we probably share with you as well. We want to contribute to treat the untreatable, to cure the sick, and ultimately make a change. One can do that by means of regenerative medicine. There you replace dead and dysfunctional cells with new cells that you have expanded ex vivo in the dish. And with laminins you can actually make this in vitro niche and restore lost function. With laminins you can recreate the authentic cell niche in vitro, and therefore your cells will also maintain their phenotype. And that is whether they are pluripotent, or if they are dopaminergic, or if you want them to become cardiomyocytes, or hepatocytes, or even pancreatic islets. So how did all this start? Now I want to take a side story from the story. There was a professor at the Karolinska Institute called Carl Tryggvason, and he's the scientific founder of BioLamina. He wanted to differentiate pluripotent PSCs with the help of different laminin isoforms, and he did not quite succeed. He complained to a friend of him, who was also a professor at the Karolinska, Outi Hovata, and she was working on maintaining pluripotent cells in vitro. And when she heard about these problems, she said, wow, that's fantastic. And that's how it all started, that Outi Hovata started using laminin 521 for cultivating PSCs. And what they saw was that the cells could be plated even as single cells without adding ROCK inhibitor, and they would attach well to the flask, they would migrate, they would start dividing, forming these monolayer islands, and within just a few days you would have a plate that was full of pluripotent PSCs without any spontaneous differentiation. Moreover, they maintained the pluripotency over time in vitro by expressing typical markers and also having the ability to generate the 3 germ layers and also forming teratomas after in vivo injections. And most importantly, they were genetically stable, which has been shown with many PSC lines by now. This effect can be partly explained if you look at what the cells themselves produce. They do not produce vitronectin, they do not produce collagen, they do not produce fibronectin. The PSCs in culture produce laminin 5-1. And also, if you knock out the alpha-5 gene from these cells, you can see that they will die on all other substrates, but if you place them on laminin 5-1, the cells are rescued. This has been shown by Laperle in 2015. And why? Why does this all work? How do I bring this back to where we started about the niches in the body? Well, if one looks where these cells are in the body, they are present in the blastocyst, as you can see here, the inner cell mass during development. And there, these cells express laminin 5-1-1 and 5-2-1. So in essence, we are mimicking exactly the environment which they encounter in vivo, also in vitro. There's another laminin actually present this early on. It's in the Reichert membrane and it's laminin 1-1-1. They are actually needed, both of them. They work in concert with each other. However, laminin 5-1 promotes the pluripotency at this stage, whereas laminin 1-1-1 is needed for the differentiation of these cells. So far, we only looked into one niche, the niche for pluripotent stem cells. However, the same reasoning can also be applied to all other niches in the body. And now, I would like to dig deeper into using BioLamina laminins for developing not only better cells, more authentic cells, but also making clinically relevant protocols. And we will be using here some customer examples also. We will start by digging deeper into the brain. During development, we know that different laminin isoforms are expressed differently among the populations that make up the brain. And also in the adult, one can see different laminin isoforms. They are more condensed and specified to be around neurogenic zones, so where we have the stem cells in the brain. But as you can see from the list, there are actually quite many laminin isoforms associated with the brain. Malin Parmar and Agnete Kirkeby, as you can see down right on the slide, they actually made use of combining what they knew about the biological relevance of the laminins and also looking at their differentiation protocol. And by combining those two knowledges, they could make better cells, but also made the protocol more efficient and at the same time regulatory compliant. Agnete Kirkeby and Malin Parmar are pioneers, and they are also carrying on a long heritage of pioneering work from Lund University. They are studying how to make dopaminergic progenitors and also how to use them in treating Parkinson's disease. So what they did was that they expanded their starting population of cells, this was human embryonic stem cells, and used laminin 5-1. And thereafter they shifted to laminin 1-1-1 for the targeted neural differentiation. In the end, they had increased the yield of transplantable neural progenitors by using this protocol more than 40 times. And the importance of this cannot be underscored, since that takes away one of the main bottlenecks when it comes to clinical manufacturing, namely scale up. And this work was published in Cell Stem Cell a couple of years ago, an excellent read. The next niche we'll focus on is the eye. In age-related macular degeneration, there's a dysfunction of the retinal pigment epithelium, RPE. And RPE supports visual cells in the back of the eye. The basement membrane of these RPE cells is called Bruch's membrane, And there are a few distinct laminin isoforms associated with the Bruch's membrane, which you can see in the list. Fredrik Lanner and Anders Kvanta, both researchers in Stockholm, made use of this knowledge and cultivated their HESCs on laminin 521, and then they differentiated these HESCs towards RPE using the laminins, and they also injected these cells into a preclinical eye disease model. They could then see that the cells integrated with the host cells, they formed a monolayer, they expressed suitable markers, and they were also functionally active. And the last niche that we will focus on is the pancreas. The basement membrane surrounding pancreatic islets contain laminin 411 and laminin 511-521. That is produced by the endothelial cells. And for beta cells to properly be able to produce insulin, they need to have laminin 511 and 521 present. And Carl Tryggvason, who is now working in Singapore, used this knowledge and cultivated islets, pancreatic islets from mice, monkeys, and humans on top of laminin 5s, so both 511 and 521. And the islets attached nicely. They survived for a quite long time in vitro, and not only survived, but they could also observe that the islets expanded, and it was an even expansion. So all the cells within the islets proliferated, i.e. the homeostasis was maintained. Again, this increased survival increases the lead time from islet donation to recipient transplantation, and it might even reduce the need of the number of cadaver pancreas needed. We now have, as you can see, the LN521, the MX521, and the CT521. I will get back to some of the differences later on, but let's look at functional. What are the functional difference between them? Well, if you compare the doubling rates when you plate pluripotent cells on CT521 versus LN521, you can see that there are no differences between the substrates. What you can see is that the cells will also proliferate when put on vitronectin, but to a lower extent. However, what we can see is that fibronectin does not support the maintenance of pluripotent stem cells. Furthermore, the cells will attach as single cells without the need for rocci, and they will proliferate and form the monolayer within a few days without any spontaneous differentiation. Also, the cells are pluripotent, as shown here by the expression of pluripotent markers such as TRA160, SSEA4, NANOG, and OCT3-4. So what are the differences? We provide now 3 BioLaminin 521 substrates: LN521, MAX521, and CT521. LN521 and MAX521 are both research grades, whereas the CT is obviously cell therapy grade, meant to be used in manufacturing of cell, gene, and tissue-based products. However, the MAX521 and the CT521, they share the same production cell line origin, whereas the LN521 has a separate adherent production cell line. LN521 and MAX521 share the same quality control criteria setup, whereas the CT521 have an enhanced quality control at the end, which is also supported by pharmacopoeia. So to summarize what you will get from us by buying the CT521, you will get a substrate which is designed to be used when you make cells that will go to the clinic. You will get all the documentation that you will need, and because of the nature of the laminin 521, you will also get a consistent and reliable performance of the cells, and the whole culture system is flexible and very easy to maneuver. And now to conclude the whole presentation or webinar. BioLamina believes that you have to start with good cell culture by looking at the biorelevance of your cells. You have to look at the niche in vivo and recreate it in vitro. Then we support you by giving you all the laminins you will need to take you all the way from idea to ultimately the cell product. And we offer you great value because the product can be easily translational, it's easy to use, you minimize variation, and by all this you also reduce your cost. Our best ambassadors in the end are our happy customers, and they truly are our stars, and they help us to also spread the word about the laminins. Please read more about them on our homepage. Thank you all for attending this webinar, and please read more about our cell therapy grade 521 on stemcellvision.com, and don't forget to reach out if you have any questions.